Good evening to everyone, and welcome to the seminar from Professor Malliaras from the University of Cambridge. And this talk belongs to the series of science colloquia organized yearly by our department with the monthly appointment in which experts in different fields of physics gave us the opportunity to improve our knowledge. So thanks you for coming, and I'm leaving the floor to Professor Torres to introduce the speaker.
Thank you very much, Livio. And let me say, it's a great pleasure to have George Malliaras here from the University of Cambridge. George is a good friend of mine, at least, and thank you very much for coming. So George got his BSc from the Aristotle University in Greece, and then he moved to Groningen to get his PhD, and then he stayed there as a postdoc. And then he decided he wanted to go to the US, right? So he went to Cornell, and then he stayed there until, I think, 2006, before coming back to Europe, to the École de Mines Etienne in France, to actually establish, I think, one of the first departments of bioelectronics in Europe. And he stayed there since, till 2017, when he actually came to Cambridge as the Prince Philip Professor of Technology. So the Prince Philip Professorship is actually a professorship established by Prince Philip, the late Queen's husband, actually to essentially attract eminent people to the University of Cambridge, and it's actually one of the most prestigious professorships in Cambridge. So George is a recipient of a very large amount of awards. Just to say a few, the MRS Mid-Career Researcher Award and the Blaise Pascal Award from the European Academy of Sciences. He's a fellow of the Royal Society, and he's going to talk about technology for bioelectronic analysis.
Thank you very much, Felice. It's great to see you again, my former colleague, and now at Imperial and at Catania. It's wonderful to be here. It's my 5th time, I think, in Sicily. I always enjoy being here, both on business and on vacation. It's a wonderful place. I come from Greece, as Felice mentioned, so I feel very much at home here. We'll talk about technology for bioelectronic medicine, and I'll give you an overview of the issues, some of them scientific, some of them more technology related. Hopefully try to cover a broad area. And I will explain what bioelectronic medicine is and why it needs new technology. So let me start with this. This is a patient who suffers from a neurological condition called tremor. It's characterized by involuntary motion of the extremities that makes life very difficult to live. And this is the same patient here, implanted with a device that presents electrodes in a part of the brain called the subthalamic nucleus. And through electrical stimulation of that part of the brain, the symptoms ameliorate dramatically with a flick of a switch. It's not perfect. It's not a cure, because if you switch the device off, the symptoms return immediately. But it is a treatment of the symptoms that's quite effective. This technology is called deep brain stimulation, and it's one of the many facets of bioelectronic medicine. Now, as a field, the use of electricity to act on humans goes back to the ancient world, where electric fish was used to treat people with migraines. But it was an Italian, Luigi Galvani, in the 18th century, who did the pioneering experiments that led to having the field placed on a scientific basis. And this is the famous experiment with a dead frog, application of electricity. The legs got to twitch. And this gave rise to lots of theories about animal electricity, the nature of the nervous system. And 200 years later, there were already medical devices. This is a cardiac pacemaker that was used by people with hard arrhythmias. And at that time, this is in mid-1950s, the electronics and batteries were external. So the patient had to carry with him this card of electronics. In 1958 was the first fully implanted cardiac pacemaker that started the modern era of bioelectronic medicine. So if we start the clock at late 50s with a cardiac pacemaker, this presents electrodes on the heart. This is the location of intervention. And it started the era of modern bioelectronic devices. 10 years later, there were cochlear implants for treating profound deafness. Here, the location of the intervention was the inner ear. I'll show you a device in a moment. Then there were spinal cord stimulators for people who suffer from chronic pain. Then a couple of other devices, 80s, 90s, and then in 2000, the brain came to the forefront with deep brain stimulation for neurological and neuropsychiatric disorders. For example, severe forms of depression, obsessive compulsive behavior can now be treated through electrical stimulation. And then the floodgates opened up and many more devices became available. This is not a comprehensive list. The latest and greatest is peripheral nerve stimulators that help people with diseases that go beyond the neurological spectrum and into autoimmune diseases, for example, Crohn's disease, type 1 diabetes, rheumatoid arthritis are all believed to be treatable through electrical stimulation. And this has gotten important enough that it attracted the interest of Google's parent company, Alphabet, who collaborated with GlaxoSmithKline, a massive pharmaceutical, to launch a company called Galvani Bioelectronics in the UK, who envision a future where disease will be treated through miniaturized implants. And of course, since then, other prominent cases such as Elon Musk's Neuralink was created. They're already having FDA-approved devices on a couple of patients, and the field is taking off. There's a lot of hype. This is typically expressed in movies by man with bioelectronically controlled tentacles, wreaking havoc in our civilization. The clinical reality is more what you see here. So this is a boy who was born deaf and is wearing a cochlear implant. This is a device that has an external part that has a microphone that picks up the sound, then some electronics to process the sound, and an antenna that radios this information to an implant in the inner ear that stimulates the cochlea, that's the hearing organ, and gives rise to the sensation of sound. And look at the marvel in the eyes of this boy as he hears for the first time. So we're talking about life-changing technologies. Now, any medical technology comes with ethical concerns. And here, because we're acting on the brain, which is the organ that gives a sense of self, is the seat of our consciousness, those ethical concerns are compounded. For example, there are issues of agency. Is it me who did that, or is it the implant that stimulated my brain and made me do it? This is from an old movie that shows the quintessential mad scientist working in an ivory tower away from society, trying to galvanize, as it was called, or reanimate a dead corpse by applying electricity. And this is a parody of a scientist who is completely disconnected from society. How do we end up with devices like this? Is through involving all the stakeholders from the very early stages of development. So to develop a medical device, an implant that saves lives, This is a type of cardiac stimulator. It's developed by collaborating from the very early stages with patients, their advocates, ethicists, regulators, industry. If there is one thing that you retain from my talk today is that medical device development is a game of multiple stakeholders, all of whom should be consulted from very early on. So with that as an introduction, very broad one, let's talk about the limitations. This sounds very promising. You implant a little device, turn it on, stimulate the different locations in the body, and treat the symptoms of disease. Why don't we see this deployed to patients at scale? There are a couple of reasons. Some are fundamental. We do not understand how the brain works to the point where we can find the target location for interventions. The human brain is a very complex machine. It has over 80 billion neurons, each one connected to as many as 1,000 other neurons. So the number of connections is astronomical. And although we understand how two neurons communicate with each other, we know that neurons, for example, have processes that are called dendrites. They accumulate information from their environment. That information gets compiled in the soma, the body of the neuron. And then the output of that computation is an action potential, as it's called, or a voltage pulse that travels down a long process, which is called the axon. And when it arrives to the terminus, it can secrete neurotransmitter. That then, these are some biochemical molecules that can either stimulate or inhibit, so either excite or inhibit the postsynaptic neuron. These are phenomena at the molecular level that we understand reasonably well, but we do not understand how we go from this neuronal phenomena to macroscopic behavior. Neuroscientists are trying to solve this puzzle by putting electrodes at different locations and probing the brain at different spatial scales. For example, implantable electrodes that penetrate into the brain can measure the activities of single neurons. Electrodes placed on the cortex, this is called electroencephalography, stereotactic electroencephalography, this is called electrocorticography. Here you probe larger assembles of neurons, and then electrodes placed on the scalp outside the brain, electroencephalography. Those can measure even larger clusters of neurons, actually networks of networks. And through these techniques, they try to get a connection between neuronal behavior and macroscopic organism behavior. The opposite is also not understood. What is the mechanism of neuromodulation? Why, when we stimulate the brain, we change behavior? We stop tremor, we stop epileptic seizures. It's not understood. But there are also technological limitations. The way we manufacture these devices, it's not scalable. And here there is a lot of materials physics that comes to play, as I will explain in a moment. These devices that go into the brain are highly invasive and they require multiple surgeries, sometimes to adjust their position. And the environment in vivo is hostile to electronics. So the central theme of the talk is that new technologies are needed to address these limitations and enable bioelectronic medicine to reach more people. So let's talk about the manufacturing first of implants. This is a current snapshot of different implants out there in the field. So there are implants that go on the head, on body, arms, legs, each one treating a particular condition by having electrodes placed at different locations of the body. And by and large, all these implants look like this. There is a little box. This is called the implantable pulse generator. It has about 2/3 of this volume is a battery, and about 1/3 is electronics that creates current pulses. And then this is connected through leads to one or two electrode arrays with, in this case, 4 electrodes each. This is a deep brain stimulation system, and it has 2 electrode arrays because you place one in each hemisphere of the brain. So again, these are the electrodes placed in the subthalamic nucleus. And then you have the leads, one level of connectivity here under the skin. These are these connectors. And then leads to the implantable pulse generator, which is placed under the skin, typically here on the chest. Now, all these electrodes in all of these devices look very similar. This is an electrode that is used, an electrode array in cochlear implants. It goes into the hearing organ, which looks like a snail. That's hence the shape of the device. These are spinal cord stimulators, and they all share a very similar geometry. They have platinum iridium electrodes. These are made out of chunks. You have a sheet of platinum iridium that you would cut, and then you encapsulate them in an insulator, which is typically a polyurethane or a silicone. And those are made by hand. Surprisingly enough, all implants in the clinic today are made by people working through microscopes with tweezers, putting little things together. This is the technology of watchmaking. The mechanical watches were made like this, and implants are made like this today. Coming from a microelectronics background, you find this very surprising because if you go to ST Microelectronics, for example, you will see they have clean rooms full of equipment that runs in an automatic fashion using processes such as film deposition, lithography, etching, and producing objects of exceptional precision, reproducibility, and ever decreasing cost according to Moore's law, more functionality at the same level of cost. So this technology is slowly making its way into implants. This is one example here. This is an implant that is placed on a very thin and flexible sheet of plastic, which is only 4 microns thick, so thinner than, about 20 times thinner than the width of a human hair. And it has electrodes that are microfabricated to the order of tens of microns. This is a critical dimension because that is the size of a soma of the body of a neuron. So when you make electrodes the same size as neurons, then you can communicate with individual neurons. But microfabrication is one part of the puzzle. There is some interesting materials physics that comes to play that needs to be employed to allow to enable microfabrication. And the argument goes as follows. When neurons fire in the brain, they put ions in the brain in motion. So if you have an ion in the brain close to your electrode, that ion will move about as it couples to the activity of different neurons. So it will sum up the spatiotemporal disturbances caused by all the neurons. What an electrode will do, it will transduce this motion through coupling of electronic charge with these ions. So inside your metal electrode, there are electrons that electrostatically couple with ions. When this ion moves about, this electron will move about, and that creates a displacement current that you measure in the outside circuit. That is the basic mechanism of transduction between electronics and biology. Similarly, when you stimulate, what you do is you pump an electronic current into your metal electrode that pushes ions to come closer to neurons. They charge the neuronal membrane. and the neurons then get to fire action potentials. The problem with this configuration is that the interaction between ionics and electronics is fundamentally limited by what is called the double layer capacitance. This is the property that describes the interaction between a sheet of ions and a sheet of electrons near the surface of the metal. They form a capacitor which has a capacitance, regardless of what material you use, of the order of 1 to 10 microfarad per square centimeter. How can you overcome this? How can you increase the interaction between electrons and ions? You can do this by having mixed conductors. Mixed conductors are materials where both electronic and ionic carriers can move. This means that ions from the biological environment can penetrate into these materials and couple with electronic charges throughout the volume. This allows you to reach capacitances that largely exceed this double layer limit. So the interaction between the worlds of electronics and ionics is now volumetric and is much stronger. This can be discussed in terms of an electrochemical impedance, which describes the mismatch between electronics and ionics. And by going from a flat electrode to a volumetric electrode where ions can penetrate into the volume, you lower the impedance. But these are details. So how is this done practically? It's done by using organic materials that are called conducting polymers. This is one example. We'll examine this in more detail in a moment. Imagine this for the moment as a spongy conductor where ions from an electrolyte can penetrate into its volume and interact with electronic charge. This manifests itself by a capacitance that is scaling not as the area of the electrode, but as the whole volume of the electrode. So the thicker you make your electrode, the higher the capacitance, the higher the interaction between ionic and electronic charges. So that is the basic physics of the transduction process. So, and it enables us to miniaturize electrodes. This is a clinical electrode that is used today to map the brain. This is made by hand. It has very large electrodes. You see the electrodes are of the order of several millimeters in diameter. This is a procedure that is used before any operation in the brain. They have to map the brain to know where to cut and where not to cut. And by using microfabrication together with this conducting polymer coatings, we can go to hundreds of electrodes in the same space, having a resolution now that is commensurate with recording single neurons. So by using this piece of science, by being able to mix electronic and ionic charges in the volume of an organic material, we can go from resolutions of several millimeters to single neuron resolution. Now, let's talk a little bit about the physics of this process. Carbon as a semiconductor. If you have conjugated chains of carbon, then they're semiconducting. How do we understand this? If you take ethylene, which is the simplest conjugated molecule, is bonded in a fashion where you have sp2 bonding between the carbon atoms and carbon and hydrogen. So this defines a planar structure. You can imagine this being the structure of the molecule with the 2 carbons here and the 4 hydrogens at the corners. And over and under, you have, so this leaves one electron per carbon atom in a pz orbital, but these two orbitals overlap, allowing these electrons to delocalize throughout the length of the molecule. So you have an electron cloud above and below the length of the molecule. Now, so this type of bonding gives rise to electron delocalization. Electrons that are delocalized in a nanoscale box exhibit particle in a box behavior. So they have quantized levels. And if you start filling up with electrons, you end up having a band gap that scales with basically inversely proportional with the length of the molecule. So that tells you that you can engineer these conjugated molecules to have an energy gap between the highest occupied and the lowest unoccupied molecular orbital that is in the semiconductor range. So a conjugated molecule is a semiconductor. This is shown here. This is a polythiophene, and it shows this principle of conjugation, the alternation between single-double, single-double, single-double bonds. This gives rise to this sp2 bonding, leaving one electron per carbon atom in a pz orbital. So this is a semiconducting polymer. But in this case, when it's brought into proximity with a macromolecule that contains anions, then it gets doped p-type. So what happens is a hole is created in this electronic structure, and that hole can move up and down this molecule and jump from one molecule to another, giving rise to macroscopic conductivity. In the language of chemistry, this chain is oxidized and you create a whole polaron or a positive polaron that can move up and down the chain. So this is a prototypical conducting polymer called P.PSS, where P dot is the semiconductor that is degenerately doped p-type through the sulfonate anions that are immobilized on this PSS polystyrene sulfonate chain. Now, how do we study this phenomena? We study them using a configuration called the moving front experiment. And this is the beauty of organic electronics, that this phenomena you can see with your eyes. So we take this material P.PSS and we deposit it on a glass substrate and then cap it with a layer of material such as SU8 that cannot be penetrated by ions. And then with a process of etching, we expose one edge to electrolyte and then have a back contact on the other side. Now, P.PSS, as I mentioned earlier, it's degenerately doped. So it has holes that can move about, and it has these sulfonate anions that are fixed in space. These are the dopants that are fixed because they're chemically linked to this polymer chain. But when we apply a potential, as shown here, so negative with respect to what we apply on the electrolyte, we pull the holes towards this side. And at the same time, we inject cations, positive ions, into the P.PSS. So what happens is as cations come in here, they compensate the anions, and the holes recede towards the back contact. So you see a de-doping front that moves from the left to the right as time goes by. And you see a de-doping front because the color changes. So here, when the holes move away, the color changes slightly because these materials are electrochromic. So this is a picture of the sample taken from top. And we switch on the voltage, and what you see is a de-doping front cross over the material. So you use a little stopwatch to time the time it takes for ions to go from this side to that side, and that gives you the ion mobility. You can analyze this data quantitatively. You can look at the transmission through the sample as a function of time, and you see this de-doping front move through the sample. This contains an enormous amount of information, not just mobility, but can give you more information about the character of transport. Now, how many of you have heard of organic light-emitting diodes? These are devices that are very common today on cell phones, on big displays. And the physics of these devices was being worked out in the late 80s, early 90s. And what was crucial in understanding how these devices work was an energy level diagram. So an organic light-emitting diode is the simplest configuration, a stack that consists of two metallic electrodes sandwiching an organic semiconductor. So the organic semiconductor is represented by the highest occupied molecular orbital, which is the edge of the valence band, the lowest unoccupied molecular orbital, which is the edge of the conduction band, and the two electrodes are represented by the Fermi level. One has a high work function, and the other has a low work function. And these devices work as follows. If you apply an electric field between the two electrodes, then electronic charges, electrons, get injected from the low work function metal into the conduction band, holes from the high work function metal into the valence band. These charges move towards opposite directions. They find each other in the bulk of the material, recombine, and light gets emitted. Why is this important? Because by modeling these processes, you can tell, for example, where, in which location inside the sample, electrons and holes will get together and recombine. You want to push that away from metal electrodes and all that. In the general case, this is called the bipolar injection problem, and it can be solved numerically. And it was solved numerically in the late 90s to understand light-emitting diodes. Now, why did I start telling you about that? Because the exact same set of knowledge is now applied these days to understand the processes of electrochemical doping. This is treated like a device physics problem, where you have a polymer. It's in contact with a metal electrode on one side and in contact with an electrolyte on the other side. So if we want to dope this polymer electrochemically, what we would do is we would apply a potential, and then from the electrolyte, we will inject, for example, anions. They will go in. From the metal, we will inject holes. They will go in, and then they will combine at some point, compensating electrically each other, and the material is doped. This is the process of electrochemical doping. So these are the elementary steps, very similar to a light-emitting diode. You have steps of injection, transport, and then compensation as opposed to recombination. And these, of course, are interdependent. They're correlated through changes in the local electric field and band structure. So typically, you would solve drift diffusion equations to get the physics of doping. And then here is how the result could look. This is, again, the moving front experiment. Here we do it for a polymer which is not doped. It's pristine. And what we're going to do is we have contact with a metal on one side and an electrolyte on the other. So we apply an electric field. Ions will come in through here. Holes will come in through here, and they will meet somewhere in the sample. The question is where? And until very recently, that question was not answered. And there is a different answer for different classes of materials. So here is how it goes for this material. You apply the potential, and what you see is that the moving front starts from here, moves this way, and then moves that way back. Look at this again. So you see the moving front move this way and then that way. This is more complicated than the previous case I've shown you. So what happens in this case is we first inject ions. They move faster than holes, which is very surprising. And they come and accumulate at this side of the interface. This pulls in holes because it creates high electric fields. That's what you see in the moving front direction. And after enough holes have gotten into the sample, then the moving front direction will reverse. Why is this? This has to do with the nature of transport in disorder materials. Holes, when they first get injected, they live at the very bottom of the transport manifold, where the density of states is very low. As a result, their mobility is very poor, and it can be slower than the mobility of ions. However, as we start filling up these low-lying states, then the Fermi level moves at a higher density of states, and then ions, then they become, holes become faster than ions. And of course, this is supported by measurements of mobility as a function of carrier density. We can do this in transistors. And you see that the hole mobility at high charge densities is very large, can be quite large, while at the low charge densities plummets by orders of magnitude. One last thing, just to show you that the physics of transport of these materials is truly intriguing. Here we're measuring the ratio of current carried by drift and by diffusion in these materials. So we're doing a de-doping experiment in P.PSS, and we look at what fraction of the current is by drift and what by diffusion. For cations, the majority of current is carried by drift. The ratio is over 10. And then it comes down to 1. When the cations reach the end of the sample, we have steady state, which is defined by drift equals diffusion. So this becomes 1. Holes, on the other hand, seem to follow a very different law, where they still move by drift. The majority of the current is because of drift, but their diffusive component is much higher. This is called anomalous diffusion. It does not obey the Einstein diffusion equation, which is something that often happens in amorphous material. Now, enough with the fundamentals. Let's talk about devices now. And I'll show you an example of how you can use these microfabricated devices to develop a new device which aims to bypass spinal cord injury. So the way our motor system works is the brain would send a command for motion. That motion will, that command will come down the axon of a neuron. It will stimulate a lower motor neuron which lives in the spinal cord. That command will then be propagated through peripheral nerve to muscle to get it to contract and move. At the same time, sensory receptors in the body will understand that the muscle has moved and convey this information through peripheral nerves, through the dorsal root ganglion of the spinal cord. These are sensory neurons that live in here, and up towards the brain. And there is also a local reflex circuit here. Now, when the spinal cord is injured, those pathways are disrupted. Information from the brain doesn't reach the muscle, and people end up with a mobility deficit. So how can we develop an interface that helps restore mobility? These are examples of very small, very flexible arrays of electrodes that can be wrapped circumferentially on the spinal cord, as shown here. So this is the spinal cord, and this array is wrapped 360 around it. That allows us to study information transmission from the brain to muscles and vice versa. For example, if we stimulate a peripheral nerve and create a signal that goes towards the brain, this is called somatosensory evoked potential. And we're measuring signals that emanate from the periphery towards the brain. If we look around the spinal cord, we see that every time we stimulate here, we pick up activity here. This is the back of the spinal cord, the dorsal part of the spinal cord. On the other hand, when we stimulate the brain to send a motor command towards muscles for movement, we measure this at the ventrolateral part of the spinal cord. This is the part of the spinal cord that's on the side and towards the belly. So we can map these signals, and then we can use the electrodes around the spinal cord to stimulate with these signals and elicit motion. So here are different electrodes placed around the spinal cord, and we're stimulating here the ventrolateral area. And by stimulating from the different electrodes, we can produce different motions in the animal. So then what we can do is we can put two arrays of electrodes, one above, one below the injury. This one records information from the brain for motor command. These motor commands do not transmit through the injury, so the animal would not be able to move its legs, but we catch it on its way down, and then we use this information to stimulate below the injury and elicit that motion. And this is shown here. So these are the recordings that we get at a particular location here. And then these are the effects of stimulation. So we take this recording, we bring it here, we stimulate, and then we measure with electromyography the movement of the arm, the leg of the animal, and we see that we elicit a movement. We can also do kinematics to look at the angle of movement and all that. That only happens when this interface is on. If we switch off this interface, then we can measure this command as it comes down from the brain, but it doesn't transmit through the injured spinal cord. So this is a very small step towards making a bioelectronic bypass or passing information across an injury site. Now, in the last few minutes, I want to talk about the interface with soft robotics. This is an emerging area. What is soft robotics? This is an example from George Whiteside's group where you have an object made of silicone rubber and you pneumatically actuate it by using fluids to change shape. As a result of the change of shape, it moves forward, but the important thing is the change of shape here. So can we use this concept in bioelectronics? And the application scenario is as follows. In spinal cord stimulation, which is an established neuromodulation technique used to help people with chronic pain, there are two types of electrodes. One is called the percutaneous array. This is a linear array of electrodes that gets placed on the spinal cord through the space between vertebrae. You just jab a needle here and push that array up the spinal cord. This is a procedure that is similar to an epidural injection that is given to women during childbirth, or a spinal tap to remove a cerebrospinal fluid. So a rather minimally invasive operation. So easy to place. Unfortunately, these electrodes often move inside the body and they lose efficacy. A better technology is this paddle electrode, which is two-dimensional. But to place that on the spinal cord, you need a laminectomy. A neurosurgeon needs to remove part of the vertebrae to place that electrode in. And this means the patient has to stay in the hospital for a couple of days. So it's more expensive to perform, but it's a much better way to do it. So with concepts from soft robotics, you can have the best of both worlds. You can make electrodes that are very flexible and have embedded microfluidics. So you can roll them up to look like a percutaneous array, put them in the body with a minimally invasive procedure, and then open them up. So this is shown here, that you use a needle to do a lumbar puncture, access the space in the spinal cord, place the electrode in, and then once it's in location, you open it up and becomes a puddle. So this was validated in human cadavers. This is the injection site. This is the electrode being placed, and then it's going to move up. There is a fluoroscopy set up here to image that expansion. And once it's up, then the surgeon can pressurize it with a syringe to deploy it. So you see this here. This is the electrode placed on the spinal cord. There are two X-ray markers that coincide when the electrode is rolled up and then begin to separate when you deploy it. And then you can go and verify the placement. So this is the injection site. This is the laminectomy here to open up and see that the electrode hugs the spinal cord very nicely. This one here and this one there are the two X-ray markers that separate it. Another device you can do for with soft robotics is an intraoperative recorder. So when a surgeon operates in an area that has a lot of nerves, they monitor these nerves using typically handheld probes, which are quite dangerous because these nerves can be very sensitive, and the mere act of touching them can lead to injury. So there is a lot of interest in developing electrodes that are very soft, very thin, that can go and wrap around the nerve, as shown here, to monitor it during an operation. But most of these concepts are permanent placement of electrode. There is need for temporary placement of electrode, which remains unsatisfied. So here again, we resort to conducting polymers. There is a concept called artificial muscles, where if you have a bilayer between a metal and a conducting polymer, and you apply a potential between this and an electrolyte, ions from the electrolyte will come in and swell this polymer and cause the bilayer to curve in this way. Or if you pull the ions out, then they will go the other way. So this is shown here, and it can be quite a dramatic effect. So this is by applying just a small voltage pulse, you can get this to open and close. You can also engineer the type of motion and go to helicoidal motions by patterning the conducting polymer at a slant with the axis of the implant. And then you can elicit this helicoidal rolling motions, and you can get it to wrap around nerves. So this is shown here. This is the electrode design. It has this slanted conducting polymer actuators that give the helicoidal motion, and also microelectrodes that can measure nerve traffic. So when you put this on a nerve, by itself, it doesn't perturb the nerve. It doesn't cause any neural activity. While if you gently press the paw of the animal and cause somatosensory, somatosensory evoked potentials to go towards the brain, you can catch them here, maintaining, ensuring that you maintain nerve health during an operation. So an example of a technology where conducting polymers again can give rise to needed medical technology. So with that, I hope I covered a lot of area, something for everyone. I hope I gave you a flavor of what's to come. Bioelectronic medicine represents a new way to treat disease through electrical stimulation. Using microfabrication, we enable scalable implant manufacturing. This is something that will come in the future. And here you need something in addition to just lithography and etching. You need materials that offer a low impedance or a volumetric coupling between ionic and electronic charges. These are the true heroes. This is what enables miniaturization. Thin film electrodes with circumferential recording and stimulation capabilities enable a spinal cord interface bypass. This will be for many years in development. We're now seeing just very crude motion, and we need to understand a bit how the spinal cord transports information in order to make it more refined. And then the interface with soft robotics enables shape-actuated electrodes. The pneumatic one will be in patients soon. There is a company behind it that's pursuing translation. The electromechanical one that wraps around the nerve still is at the early stages of translation. So with that, let me thank my group, which is co-led by a Neapolitan neurosurgeon and a Spanish neuroscientist, a collection of anything from physicists, chemists to neurosurgery. It's a very interdisciplinary group, as it needs to be. And then collaborations with people at the university and outside, funding agencies. Thank you for your attention, and thank you for the kind invitation.